Hello everyone. So we are going to continue with the third part of our chapter, which is photosynthesis in higher plants. So till now we have covered the portion till the experiments, I guess. Yes. Okay. So in today's class, we are going to read about where the photosynthesis takes place. So let's get into the video. Okay. So let's start. You would of course answer in the green leaf of the chloroplast. Okay, so we know that where the process of photosynthesis takes place, we are going to learn it. It takes place in the mesophyll cells. Wait, something happened in the mesophyll cells. In the mesophyll cells of the leaves, okay, and have a large number of chloroplasts. And usually the chloroplasts align themselves along the walls of the mesophyll cells. Like if it is the wall of mesophyll cells, then the chloroplasts will be going to align at the walls of it. Okay, so where it is happened in the mesophyll cells in the leaves, which has a large number of chloroplasts and which are majorly aligning along the walls of the mesophyll cells, such that they'll get the optimum quantity of incident light. Like for example, from here we are getting the light. It is the incident light, okay, and here the leaf. It is a leaf. It is a mesophyll cell, okay. Well, let me explain you. Okay, so let's suppose, let me make a good diagram for you. Suppose this is a sun, okay. Don't take my drawing seriously. And this is a leaf, okay. This is a leaf, okay. And we have light. From here, from the sun, we are getting the incident light on the leaf. This is mesophyll cell, okay. This is our mesophyll cell. This is mesophyll cell, okay, which has the chloroplast aligned at the wall. So it will be easier if it is at the lying at the wall of the mesophyll cell. It will become easier for it to trap the sunlight. No, so that's why it is aligned in the walls of mesophyll cells. Okay. Next, what is written? Next written is, okay, and now we are going to learn the structure of the chloroplast. Okay, let's talk about this diagram. This diagram will make you everything clear. This is the outer membrane. This is the outer membrane. Okay, this is the outer membrane. This is the inner membrane. Inner membrane, outer membrane. This is stoma lamellae. This is stoma lamellae. Okay, look, yeah, these structures, like this structure, this is made up of, this is grana. What is it? It is grana, and grana is made up of thylakoids. Like grana is this the group of this like structure na. So the group of thylakoid like structure, thylakoid group of thylakoid makes a grana. So a group of thylakoid will make one grana, okay, and one or more grana are attached together by a structure which is called as what? Which is called as stoma lamellae. Stoma lamellae, okay. And this is our stoma, this is our stoma, this is our stoma, okay. And this is ribosome, this is starch granule, this is the lipid droplet. These droplets are lipid droplets, okay. So this was the structure. This is the electron microgram. Please take care. This is the electron micrograph, okay. of chloroplast. Now let's study what is written here. It is written that we have studied the structure of chloroplast. No, in the class 8, but we have already studied in the class 11 also. So within the chloroplast, there is a membrane system which is called as grana. And grana, two granas or more than two granas are attached by the stromal lamellae and the matrix stroma we have. Like this is the chloroplast. Here it is stroma only. Okay, stroma. It is the matrix. Okay, and there is a clear division of labor within the chloroplast, means every part has their individual work to do, so there is a division of labor. And the membrane system is responsible for the trapping only, because the membrane can only trap the sunlight, no, so the membrane can only trap the sunlight. From the membrane it will move inside, na, so the membrane is a primary acceptor or receptor of the sunlight. So the membrane is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. Stroma may enzymatic reaction synthesize sugar, which is turn forms starch. Okay, and the former set of reactions, since they are directly, like if we are taking the light directly from the sunlight is driven, then it's called a light reaction. It is a photochemical reaction. Later the light is not directly supplied to the other parts but indirectly supplied, but it's also indirectly indirectly needs sunlight. Okay, it indirectly needs sunlight, so it is not directly taking the but dependent on the products of light reaction, like the products which are formed from the light reaction, the dark reaction will depend on these products. Okay, okay, and these are dependent on the product of light reaction like NADPH and ATP. So this is called as dark reaction, or we can say carbon reaction, we can also say it. And it should not be happen that you are thinking light reaction which takes place in the presence of light and dark reaction which is done in the andhera. No, it's not like that. Dark reaction is said to be dark because it's not directly dependent on the light but surely dependent on the products of the light reaction. Okay, and we have discussed this. Now let's talk about this. How many types of pigments are involved in the photosynthesis? Let's first discuss about this. Okay, what is it saying? This is chlorophyll A, B. This is carotenoids. This is chlorophyll A. Chlorophyll A is going like this. Okay, chlorophyll B is going like this. Okay, chlorophyll carotenoids is going like this. Okay, so what is it trying to say? It is trying to show the absorption of light by the pigments. Look, we have some pigments, like we have chlorophyll A, which looks like bright or blue green. In the chromatogram we have chlorophyll P, whose color is yellow green, then xanthophylls, which is yellow, and we have carotenoids, which is yellow to yellow orange. Okay, you have to learn the colors also. Pigments, what are pigments? Pigments have the ability, these are those substances which have the ability to absorb the light. They have the ability to absorb the light at specific wavelengths of different wavelengths. Okay, and chlorophyll A pigment shows the ability to absorb light of different wavelengths. Which pigment? Chlorophyll A. Chlorophyll A has the ability to absorb the different wavelength. Okay, chlorophyll A, keep it in your mind. And this is also written that we are also familiar that the wavelength of visible spectrum of light is VIBGYOR. VIBGYOR means violet, indigo, blue, yellow, orange, and red. Okay. And it is also saying that the maximum absorption of the wavelengths, the specific wavelengths, was done by what? Our chlorophyll A. Like in the blue region and in the red region it happens. It shows the higher rate of the photosynthesis. Okay, which region? Red and blue region. Okay, next page. These graph together shows that the most of the photosynthesis takes place in the blue and the red region of the spectrum. Some photosynthesis does not take place at other wavelengths of the visible spectrum. And let us now see that how it is happening. Though chlorophyll is a major pigment responsible for the trapping light, but other thylakoid membranes like for chlorophyll B, xanthophylls, and carotenoids. Like we have what? We had what? We had four pigments, no? We had chlorophyll A, chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. We had these many pigments, no? So chlorophyll A is the maximum absorber. But what's the role of B, xanthophyle, or carotenoid in it? As it is not doing like, so the maximum work is done by chlorophyll A, no? So what's the function of them? So it is writing that like the chlorophyll A is the, it's not written A. Chlorophyll A is the major pigment responsible for the trapping of light, but other thylakoid pigments जैसे कि chlorophyll B, xanthophylls, carotenoids are called as accessory pigments because they also absorb some light and transfer the energy of chlorophyll A. They are not directly doing, but they are helping the chlorophyll A as well. So they also have some function to do. That's why these are accessory pigments. Indeed, they are not capable and not only unable to a wider range of, they also help in what they, a very important role of them is written here. They not only enable a wider range of wavelength of incoming light to be utilized for photosynthesis, but also protects the chlorophyll A from getting photooxidation because because the photosynthesis is going on now, and there is like there could be a chance of that chlorophyll A will get oxidized by the photosynthesis, by the sunlight, so it protects it from that also. Next we are going to talk about what is light reaction. Light reaction, this is very simple guys, very simple. Okay, so let's now discuss this. What is it saying? It's very simple guys. What is it written? These are the pigments, these are the pigment molecules, this is the reaction center, and this is protons. Okay, these are protons. Here we have the primary acceptor, which is electron acceptor. So what happens in this? What happens when the sunlight falls on it, light falls on these pigments, then the reaction center and the electrons get excited. Electrons gets excited, and these are when then accepted by our primary acceptor. Okay, so this is what our basic light harvesting complex is. Okay, so let's read what is it writing. It is written that the light reaction, which are called as photochemical phase, includes the light absorption, water splitting, oxygen release and the formation of high energy chemical intermediates, which are ATP and NADPH. So several protein complexes are involved in this process. Like the pigments are organized in two discrete photochemical light harvesting complex within the PS1 and PS2. We have two PS, photosystem 1 and photosystem 2. Okay, this these are written on the basis of their like discoveries. It's not like you will think that PS1 will act first and then PS2 act. No, PS2 takes happens first, then the PS1 takes place. So it's written first because PS1 is discovered first, but that doesn't mean that it will going to be act first. No, PS2 will act first. These are named in the sequence of their discovery, not in the sequence of function during the light reaction. LHS are made up of hundreds of pigment molecules which are bound to proteins. Each photosystem have all the pigments except one, which is chlorophyll A, forming the light harvesting system, which is antennae. It forms antennae, and these pigments help in the to make the photosynthesis more efficient by absorbing the different wavelengths of light, and the single chlorophyll A molecule forms a reaction center here. Okay, reaction center is different in both the PS. This is the difference. This is the difference between both the PS. The first PS, which is PS1, which acts later after the PS2. Okay, it has the absorption peak. It has the absorption peak of 700, okay, nanometer, and the PS2 which acts first before the PS1 has the peak of 680 nm. So this is the main difference between them. Let's talk about the electron transport now. The photosystem 2 reaction center absorbs the 680 nm wavelength of red light, causing the electron to become excited, and when they will get excited they will jump into an orbit and further from the atomic nucleus. Then the electrons are picked by our electron acceptor, which then passes them to the electron transport system, which has cyto cytochromes, okay, which has cytochromes. The movement of electrons is downhill, like it happens like light is fallen here. Here it will get excited, here it will accept it, then it will go to the electron transport system, then it will fall on the first PS, and it will get excited again, then electron will accept it, and yes, this is Z scheme. You can see this is looking like Z scheme, so it is also known as the Z scheme. Okay, from here if we'll see it will look like Z scheme, but here it is looking like N, but from here it is looking like Z scheme, so it is named as the Z scheme. Okay, so the movement will be downhill because it is falling. Okay, and in terms of oxidation, reduction, redox potential scale, the electrons not used as they pass through the electron transport chain, but are passed on to the pigments of the PS1. From ETS, it is falling to the PS1 ka LHS, and the electrons PS1 also gets excited and receives the red light, which is of wavelength 700 nanometer, and it's transferred to another acceptor which has a greater redox potential, and these electrons are moved downhill again, and so the process continues. But it is, let me tell you, look, from here it is going to, from here it is going to here, it is here, it is here. So it is like it is a non-cyclic process, as it is no, the cycle is not getting done again and again and again. So this is a non-cyclic process. Okay, so in the last class, yes, it is written that Z scheme also we do it. Say it. Next class we are going to discuss about the splitting of water.